Hello everyone! Have you ever wondered how your body knows what to build, repair, or do at any given moment? Pause for a second and think about it. What you're about to discover today is a key process that helps answer that question. It's part of what scientists call transcription, or the synthesis of RNA from DNA template. In this lesson, we're going to focus on the first step, transcription. By the end of the video, you should be able to describe the process of transcription, explain its role in protein production, compare DNA and RNA in terms of structure and function. Let's begin with the basics. What is transcription? Transcription is the process of copying segments of DNA into messenger RNA, or mRNA. It is the first step in gene expression. Why is it important? Because mRNA carries genetic instructions from DNA in the nucleus to the ribosomes in the cytoplasm, where proteins are made. Without transcription, proteins, which are essential components of life, cannot be produced. Stages of transcription. Transcription occurs in three main stages: initiation, elongation, and termination. Initiation. It is when the RNA polymerase binds to a specific region on the DNA, called the promoter. You can think of the promoter as a starting line where transcription begins. Then we have elongation, where once bound, RNA polymerase moves along the DNA template strand, adding RNA nucleotides that are complementary to the DNA bases. So here are the base pairing rules during transcription. Adenine pairs with uracil, thymine pairs with adenine, cytosine pairs with guanine, guanine pairs with cytosine. For example, in DNA, you have TAC, GGC, which for RNA, it will produce AUG and CCG. Now we have termination. It is when the RNA polymerase reaches a stop signal on the DNA. It releases the newly formed mRNA strand. Let's identify the key components of transcription. RNA polymerase, which is the enzyme that builds the RNA strand. Promoter, the DNA sequence that signals where transcription should start. The DNA template strand, the strand of DNA being copied. The mRNA, the product of transcription, which carries the genetic message. Transcription occurs in both prokaryotic and eukaryotic cells. But there are key differences. The key differences then between the prokaryotes and eukaryotes. So prokaryotic transcription occurs in the cytoplasm, eukaryotic transcription occurs in the nucleus. Then for the RNA polymerase type, prokaryotes have one type, eukaryotes have three types, 1, 2, and 3. Then RNA processing. For prokaryotes, it is not required. For eukaryotes, it is required. This is then the splicing, capping, and the poly-A tail. In eukaryotes, the mRNA undergoes several modifications before it exits the nucleus and is used in translation. Transcription is tightly regulated. Proteins called transcription factors bind to the DNA near the promoter region to either activate or inhibit transcription. If a mutation affects the promoter or an enhancer region, it could prevent RNA polymerase from binding. This could mean the gene is never transcribed at all. For example, if the promoter of the gene is deleted by mutation, the RNA polymerase cannot attach, and the gene remains silent. Let's try a quick practice activity. Here's a DNA template strand. Pause the video now and try to transcribe this into RNA using the correct base pairing rules. The correct mRNA sequence is... Great job! Now, let's relate transcription to real-world biological functions. For wound healing, when you get a cut, your cell needs to create proteins like clotting factors and collagen. Transcription makes it possible to produce those healing proteins. Fighting a virus. So when a virus enters your body, Your immune system needs to make antibodies. Transcription enables immune cells to produce the proteins needed to fight off infection. For digesting food, every time you eat, your body makes enzymes like amylase and lipase. These enzymes are produced thanks to instructions transcribed from your DNA. Think of a situation your body might face, maybe an injury, a fever, or intense physical activity. Pause and reflect on this. How might transcription help your body respond to that situation? Write your thoughts in your notebook or the comment section. DNA has a sugar deoxyribose, whereas RNA has ribose. Then for the bases, DNA has A, T, C, G, while RNA has A, U, C, and G. The structure then of DNA is a double-stranded helix, whereas RNA is single-stranded only. Then DNA is located only in the nucleus, whereas RNA is located in both the nucleus and cytoplasm. Pause for a moment and see if you can spot one similarity and one difference between the two. Let's quickly review what you've learned. Question 1. What are the 3 stages of transcription? The answer is initiation, elongation, and termination. Question 2. Why is transcription important in protein synthesis? The answer is, it creates the mRNA instructions used to build proteins. Here's a final check of your understanding. There is a DNA strand provided. What is the correct RNA sequence? The answer is AUG, CCG, and AUU. For a follow-up question, which RNA base pairs with adenine? The answer is uracil. Today, we learned how transcription works, why it's essential for protein production, and how it connects DNA to the functioning of the entire body. Remember, even the tiniest processes, like copying a strand of RNA, are crucial to life as we know it. Thanks for watching, and keep exploring how biology makes life possible.